Right, so welcome to the Cancer and Ageing Research Programme at Queen's University of Technology. We have about 53 scientists and clinicians in our research group, and this is our main base, but we can also be found in other places in Brisbane as well, including in the city centre. So if we walk through this way, these are where most of us work, out on open plan desks. We do a lot of data analysis here, so when we get the experiments, we have to analyse the data afterwards, and that really is where a lot of the work is now spent. So we'll go into the lab if you follow me. Welcome to our lab. So this is, as I said, just one of 3 labs that we have throughout Brisbane. And this is where we do most of our wet lab work. So effectively what we're doing in here is experiments, physically doing experiments, and we're working on single proteins, single genes, and even all the way down to the atomic level. So it's a typical wet lab, maybe not what you see on television. We don't have brightly coloured bottles, but we have all our chemicals and do all the work down there. So if you follow us through, I can show you more of the lab. As you can see, we have various equipment running around the lab. In this area here, we're looking at single proteins on gels. So we can capture a single protein from the human, and we can purify it, and we can look at it on gels, and we can study the protein by doing that. So if we come through here, this is where we do a lot of our high-tech work. So this is home to our drug screening facility. So when we're looking for new targets for cancer or aging diseases such as osteoporosis and Alzheimer's, this is the main centre where we start doing this work. And what we can do here is we can screen, in our lab, we can screen up to 2 billion different compounds in one single run. Physically, in here, we can screen about 40,000 different chemicals in a single run. And that's really important because when you're looking for a new drug against a new target for cancer, you have to look for a lot of different chemicals that may work in that disease. And that's where all your drugs ultimately come from, these kind of screens. So we're probably one of the largest groups in Australia and indeed in the Southern Hemisphere looking at screening drugs. This machine here is just a black box, I guess, to you, but this is a modern microscope, what they look like. And we can screen tens of thousands of different drugs on this microscope a day. So I like to say this microscope really pushed our research forward. This microscope can do in one day what would take a person a whole year to do. So we go through, and if we come further down, we have another microscope here. It does something very similar, but in between these microscopes, this is where we do our drug screening. So that robotic arm you can see here, that will feed the microscopes with the experiments over, say, 4, 5, up to 7 days long. And so we'll be running 24 hours a day, 7 days a week, doing some of these drug screens. And you can see we also have more standard microscopes here that Thais is currently using. Thais is one of our PhD students within the lab. She comes all the way from Brazil and is spending 3 years in our lab doing her PhD. So we're a very multinational lab. We have people from South America, Europe, Asia, as well as Australia and New Zealand. So we cover everything. Science is a very international discipline. You know, a PhD is a PhD no matter where you are. This is what we call a super resolution microscope. So using certain techniques, we can see down to the individual protein level in a cell. So that could be a skin cell, or it can be a cancer cell, or it can be a bone cell. And we can open this up because it's not in use, and it doesn't look like a normal microscope. And that's because it isn't. No, it's extremely high tech. The alloys used in this are heat-stable alloys, so we can't have drift with temperature. But this machine can really see down to about 5 billionths of a meter when we use a technique called DNA paint. So this really allows us to dissect what's happening in cells, what goes wrong with aging, and how we can restore those aging phenotypes back to younger phenotypes back to our cells. So this is one of the workhorses of our lab, and as I said, it's about $1 million to purchase this one piece of equipment, so it's pretty expensive. And here we have other pieces of equipment such as this. So when we study a human protein, so we say we find a protein that we believe is driving osteoporosis, and we want to find out if we can work with that and drug that. This is our protein purification and expression facility. So all here, and this allows us to effectively purify a single human protein away from the rest of all the human proteins that are in the cell. And so we end up with only one protein, and then we can work on that protein, and that protein then goes into our drug discovery equipment, such as this. This is a machine called a dianthus. We're the first people in the Southern Hemisphere to have this machine. And basically what it allows us to do is look at single proteins and how they interact with the drug of interest. But not just once, we can do 384 experiments in one go with this piece of equipment. And we just raised money to purchase this. So this is where our fundraising comes in, and people donate pieces of equipment such as the monotemper from Dianthus. So we now have capabilities exceeding most other drug centres in this region. All right, so we're going to go to another important place in our lab, and that's where we actually grow live human cells within the lab. And we do that because we want to understand how the biology of those cells work, what goes wrong in disease, so like osteoporosis, Alzheimer's, dementia, or cancer. So we grow these cells in the lab, and we can work on these individual cells away from the complication of the whole human, and allows us to understand disease better, and allows us to then test drugs out on real cells before we go into humans. So this is one of our rooms, for example. We might go in the next room because it'll be slightly quieter. This is a typical room where we actually grow all these cells. The other room was very busy, so we'll come in this one, which is a little bit quieter. And so when we remove cells from a human to do studies on them, we do this obviously with the patient's consent. But when the cells are removed from the body, they no longer have an immune system that protects them. And therefore they're susceptible to bacteria, viruses, yeast, all these different types of infections. So we have to do it in a very clean room. So this is a clean room to start off with. It's air conditioning is separate, it's filtered. But then we work in cabinets such as these, and these cabinets have a blanket of sterile air coming up here, and you gown up, such as Ola is, and you go into these areas and you can work on the cells. And when you finish working on the cells, we can then take them and grow them in these incubators here. And I can open a door of the incubator for you to see. In this incubator, you can see all these different experiments going on. These are human cells that we're growing in there, and these are all parts of experiments. Ultimately, we're looking for new drugs to target human diseases. And these are really a workhorse areas of the lab. They're the busiest areas, as cells take a long time to grow, and most of our really relevant moving on to human experiments go to these cells before we go into humans or preclinical testing and trials. And there's some cells in there which came from patients from about the 1960s, and so they can survive a long time. Particularly cancer cells can survive an awful long time. So the next room I'm going to show you is just a basic cold room, and some -80 freezers where we store our samples. In the lab, we have our specialized equipment in these kind of rooms offset from the lab because they're a little bit noisy. So we keep the noise in there. But every piece of equipment we have they're really expensive because there's only a few of them ever made every year. And so if you come down this way, I'll show you another room. Welcome to our microscope room. So you've already seen microscopes in our other room. They're the very high-end microscopes in the other room. These are more workhorse microscopes. They're not as powerful, a lot cheaper, and generally a lot faster and easier to use. So we have a dedicated room. It has no windows because our microscopes have binoculars, so you need the dark to actually see what you're looking at down in under the microscope. Most of the things we look at are fluorescent. So we tag them with dyes, which makes them fluorescent. And so when we fire a light beam at them, they fluoresce a different color, and we capture that color on the camera. All the cameras are actually black and white, they're not color, and that's just because black and white cameras are a lot more sensitive than color cameras are. Another place we use a lot is the cold rooms. And we use cold rooms really because it slows down biological activity. So if you stick a protein in a cold room, it slows it down, its biological activity, and it doesn't degrade as quickly. And so we do a lot of experiments in the cold. These cold rooms aren't really that much different from what you'd find perhaps in a large restaurant. But you can see here we use them to store a lot of the biological agents that we use in the lab, including the food for the human cells that we grow. And also we work a lot with bacteria as ways of developing new drugs, and the bacteria are stored in here as well. The last room I'm really going to show you is, it looks like a normal freezer room. We can look through the glass there, I'll open the door in a second. These freezers are actually at -80. So when we store biological agents, we want to really slow down the biological process. So we'll put them in these -80 degrees freezers. So we open these, they're double doored, and inside there is all our reagents that we use on a weekly basis, but also things that we're storing long term. I'll shut this because it warms up really quickly. And so for instance, that's where we'll store human cells. If we're finished using them, we'll put them in those -80 freezers. And if we need to keep things even longer, what we'll actually do is we'll store them under liquid nitrogen. So we keep liquid nitrogen in the basement of this building. When we want to keep cells for years without them going off, we'll stick them in liquid nitrogen. So those cells will be in a tube, and they'll be completely submerged in liquid nitrogen, which keeps them, really stops biological activity completely. So you can keep cells for years, tens of years, in those liquid nitrogen